It can't grow in you when you're alive. What it wants to do is kill you and grow in your rotting corpse.
When we consider that 8% of our DNA came from viruses, which is actually more than the Neanderthal DNA.
And this guy just gets engulfed and eaten by the amoeba, and you can see it's figured out at some point what's happening, and you feel quite sorry for it because it's just being digested.
How is that achieved? It's utterly amazing. It's achieved by the living cell. What then happens is the whole army of enzymes go in and literally proofread the mistakes.
Your body is trained in a way that if you have cells that attack yourself, it will delete those cells.
You could carve the A, T, C, and G in granite and keep it for 10,000 years. And then in 10,000 years, type it into a sequencing machine, which we already have, and it would recreate an identical twin.
We don't have the same self when we are newborns that we do when we are 5 years old, or 10 years old, or 100 years old.
So every mind is an evolving system. Everyone has an individual evolution of our mind for structure.
I think of fantasies projected onto genes, and I think they've led to an enormous distortion of priorities in biology.
And I'm going to tell you a tale of 3 microbes to kind of give you the breadth of our interaction with microbes. One of them is lactobacillus. It lives in our guts happily and contentedly, and we are happy and contented with it because it helps our digestive system. It's what you find in yogurt. It's completely harmless, and it's one of those bacteria, along with many other in a similar category. If you take too many antibiotics, you get rid of this bacteria, and then you have all sorts of problems, diarrhea and colitis-like diseases you can have that can occur, which tells you that our balance between this organism and ourselves is well balanced. It's reasonable and it's okay. And if you upset that balance, you can get sick. So is it self or is it non-self? It's a kind of interesting one. I'll give you another bacterium, E. coli, also in the gut. Also, you find it in the soil as well. And mostly it's okay. But if you travel to another country and you meet other people's E. coli from food, from fecal contamination, then those strange E. coli's can cause very severe diarrhea and be responsible for hundreds of thousands of deaths each year. So this is one that lives happily in you, but if you meet other strains of it, it can kill you. And last year I'll give you an even odder one, a puzzle to think about for a moment. An organism called Clostridium botulinum lives in the soil. You can, if there's food contaminated with the soil, it can get into your gut. It releases a toxin, and I'm sure you've heard of this toxin. It's called botulinum, it's called Botox. It's a toxin. It's one of the most exquisitely designed toxins. It's taken millions of years of evolution to design this toxin, and it blocks your neurotransmitters, so you go into a flaccid paralysis. All your muscles go completely flaccid. It remains in your gut. It doesn't invade you. Why? Why has it evolved this terrible toxin? This toxin that is one of the most powerful toxins known to man, in order to kill you if it doesn't invade you. The reason is that it is an anaerobe, which means it's killed by air. So it can't grow in you when you're alive. What it wants to do is kill you and grow in your rotting corpse. That's its agenda. It's also in your gut. So you've got this range of bacteria that have different agendas, and that's going to be guaranteed because they're different organisms. So some we can live happily with, and we can have a natural, friendly relationship. Some not so friendly, some are beasts. And not everything is, and the beast is as natural as grandma's apple pie. It's not unnatural, it is a natural organism just like we are.
For starters, almost All the cells of every multicellular organism on Earth contain many mitochondria, tiny organelles that serve as the cell's powerhouses, generating the energy we need to survive. And they derive from free-living, single-celled organisms that were engulfed by other single cells around 2.5 billion years ago. They've transferred much of their own DNA to the host cell nucleus, and only 13 of their proteins are made by their own DNA. They are true endosymbionts, and without them, we die. But you might also say that our bodies are nothing more than an elaborate way for them to get the oxygen that they need. Such organelles are so intimately entwined with our cells that they can be considered to be part of us. And similarly, chloroplasts are a second type of endosymbiotic organelle found in plant cells that contain the photosynthetic machinery. And just this year, a third type of organelle was discovered that fixes nitrogen. And there are multiple examples of organisms that are so closely connected we can't consider them totally separate. Think of lichens formed from fungi and algae and bacteria, or the female anglerfish, one of my favourites, whose lure is provided by photosynthetic bacteria, and in which the tiny male fuses with the female to become nothing more than an irremovable appendage, sharing her bloodstream and depending upon her for nutrients. But of course, all he really is a bag of sperm so that she can have young ones. And I've not even mentioned commensalism, like the beneficial association of parrotfish and sea anemones, or parasites like ticks and lice and tapeworms, which hopefully are not on your bodies, or viruses, which lack their own protein machinery and hijack that of their host cell to reproduce. You could ask, what is an individual? And I think that's one question. But we when we consider that 8% of our DNA came from viruses, which is actually more than the Neanderthal DNA that we contain, you realise that we have evolved together with lots of other organisms. We couldn't be here if we didn't have mitochondria, for example.
The life cycle of Dictyostelium, sorry about the Latin names, you can blame Linnaeus for that, but this creature has an extraordinary life cycle. It starts with a little spore, a little bit like a seed, which has been blown through the air. It lands somewhere and happens to be water. It becomes an amoeba-like single cell that wanders around. It encounters a period of starvation, and it starts to link up with its colleagues, other Dictyostelium amoebae, that then come together to form a slug so that they can go in search of food together. And what the slug does, if it finds that it's getting a bit dry and there isn't much around to eat, it starts to grow a plant. It becomes a stalk, and at the top of the stalk there's a great big head, a global thing that contains the final spores that then get released into the atmosphere and go shooting off in the wind to start the whole process again. What agent is that? Where is the agency?
This is a quote from Derek Denton, who he died a year or two ago at the age of 97, and had thought a great deal about consciousness. And this was an example that he used to use about a fox that was caught in a trap and gnawed its own leg off to escape. And the point that he makes is the animal aware that it's held completely by its paw in the trap jaws, and does it have an intention to get free? Gnawing through its own skin, muscle and bone to set itself free is a purposive act and very probably extremely painful, you bet. At least it seems a plausible viewpoint that the animal has enough body image sense to see that its paw is part of itself that is held and its intention is to get free. It is self-aware and has a plan. Now, I am sure that not everybody in this room would agree with that. I would like to think that most would. It seems kind of obvious to me. And most people who are familiar with animals would probably see at least that level of consciousness about them. this is stentor, and it's got all these little cilia which are beating continuously, and it whirs like an electric battery going around, and this is an amoeba which is coming in, and it starts to engulf very gently and very gradually this stentor. And its friend figures out what's going on, and off it goes. It kind of zooms away and comes back for a look later on. And this guy just gets engulfed and eaten by the amoeba, and you can see it's figured out at some point what's happening, and you feel quite sorry for it because it's just being digested. But the top bit somehow wriggles itself free, and it's dead. You'd have said it was dead, but then these cilia start beating again, and they flicker to life, and they die, and they flicker to life, and finally comes back to life, pulls itself free, and off it goes. So I just wanted to show you that, because if the fox has a sense of self in escaping from a jawed trap, then did the stentor have a sense of self? So it's very easy to kind of overstretch the imagination and just say, okay, well, obviously I'm reading human emotions into a cell, which obviously has none of those things. But to any unbiased eye, it really looked like it's struggling to get free. It finally does get free, and off it goes over there. I don't think in any meaningful sense that it has an understanding that it exists, or that it has a self, or that it has an awareness or anything, but it certainly struggles to stay alive, and it acts as an entity. And those are two things that I think are perhaps quite important.
Over the course of development, your body is trained in a way that if you have cells that attack yourself, it will delete those cells, and we call this, tolerance, the mechanism of tolerance. And yes, it gets dysfunctional sometimes, and you end up with autoimmune diseases, but by and large, that is what we would define as self. And how this works is basically, when you get some kind of pathogenic invasion, for instance, it chops up this pathogens into tiny particles, and then presents it in the context of self-antigens to your body, and then your body can look at this self-antigens and say, this self-antigen and this non-self-antigen I do not recognize, and so I'm going to do something about this. And for me, that's where the basic fundamental of self versus non-self recognition comes.
Is to understand a single gene, you've got to swallow the entire genome. You've got to look at all the genes. To understand a single cell in your body, you've got to see how it interacts with everything else in your body. And now we understand that now to look at a single organism, you have to look at all of the organisms that inhabit our body, that live in our body, that we interact with. The organisms interact with each other. Mostly I work with bacteria, and you can't isolate some bacteria because they can't grow without all their mates. They need other bacteria around them to metabolize substrates that they use. So as we go from a single organ, the idea that came about, we can understand life through single genes, inspired the Human Genome Project, which hasn't really given us any advances because it had the wrong idea about genes, that genes are a kind of units like atoms, and you could understand them and then understand the whole. Now we know that isn't true. Now we know it isn't true about individuals. You can't understand a tree without understanding the fungi that it lives with, and you can't understand microbes without understanding all of their interactions, and you can't understand the atmosphere in our planet unless you know that we breathe out the oxygen that is in the air, and we breathe in the oxygen that's in the air, and it's been exhaled by plants. And similarly for nitrogen, nitrogen is fixed by microbes, and we use it to make our flesh, and then it gets back into the atmosphere. Carbon dioxide we breathe out. So our atmosphere, in comparison to our planetary sisters, is actually a part of the living system. And that's, I think, what makes us special. In order to go understand life, we have to swallow the whole world.
Perhaps we could say that genes do two quite different things. In embryology, what they do is influence phenotypes in highly complicated ways, including the ways you've just enunciated. But from an evolutionary point of view, what matters is the ones that are still here 10,000 years hence. You actually use the rather vivid image. I think you said somewhere.
Yes, 10,000 years to keep a genome in
that's
a,
right.
in a petri dish,
If you were to put a genome, suppose we put Denis's genome in a petri dish.
That's right. Yes.
And keep it going for 10,000 years. Well, it wouldn't keep going; it would decay, as you rightly say. However, the information, it could be preserved on paper. You could actually write it down in a book. You could carve the A, T, C, and G codons in granite and keep it for 10,000 years, and then in 10,000 years, type it into a sequencing machine, which we already have, and it would recreate an identical twin of Denis Noble.
No, I don't think it would.
You don't think it would?
No.
Why not?
Well, it would. It would need one. It would need an egg cell.
Of course it would. No.
Yes. I think we need the egg cell.
In 10,000 years, they will have the technology to take an egg cell.
Oh, I see. Okay, and I now therefore need to follow up on a different issue then, if I may, Richard. Yes, because you see, what it would need to be is a good self-replicator. And you won't be surprised that I disagree with you on self-replication, because I think that's a central feature. Because I think without the self-replicator, I'm not quite sure that I understand what the selfish gene idea really means. Now, let me just explain briefly why it can't be a self-replicator. The way in which that arose goes way back to the quantum mechanics pioneer, Erwin Schrödinger, who in 1942 gave lectures in the Institute of Advanced Studies in Dublin. He published it. And what he said in that book was very insightful. It was that whatever the genetic material was, whether it was DNA, protein, or whatever, it would be found to be a highly accurately reproduced molecular sequence, and he called that an aperiodic crystal. The word crystal matters there, because you see, what you say, Richard, in your books is that it replicates much as a crystal does. Now, I think that's partly true, but unfortunately not sufficiently true. Because what exactly happens, let's just go through it, and it's got to be technical for about 20 seconds or so. What actually happens is, as we all know, the double helix discovered by Watson and Crick, and Rosalind Franklin, you remember those images that were produced by, I see all the women and a few men clapping. Yes. Anyway, what Rosalind was working on, very interesting fact, was not a crystal. Her work in that critical working out of the double helix was actually on the flexible thread that actually is the DNA in a cell. You can crystallize DNA. That was done much later, but not in a living cell. Otherwise, you can never read it. Now, why is the crystal metaphor accurate to some degree, but not to sufficient degree? And it's worth just going through the figures because they're very important. What happens as the double helix unwinds is a C finds its mate because it naturally likes the other one that it likes to come in and link to it. And the same applies to the T and the G and so on. So every one of them has a mate. That's fine. Now, that is a pure chemistry thing. And you could say that's almost like a crystal forming itself, because what crystals do is that the other molecules that are in solution like to, in a lock and key fashion, go into the crystal. That's all fine. So in the same kind of way, and I think this is the reason why people like Richard say it is a self-replicator and rely on the molecular biology to say that, they're quite right up to a point. Now the question is, up to what point? In all chemical reactions, there's an energy of formation and breakage. And from that, you know how frequently it will go wrong. It's about, in the case of the nucleotides, it's about 1 in 10,000 nucleotides. Now, you might think that's fine. If you wrote a scientific article of 10,000 words, and you had only one word as a typo, you would be very pleased. But the trouble is that suffices for small viruses like coronavirus, because a mutation rate of 1 in 10,000 each time it's copied would be acceptable if you've only got, say, 10 or 20 or 30,000 as a genome length. We have got 3 billion, and the difference is around 1 million-fold. Now, how accurate is DNA replication? Obviously, that first stage, which is crystal-like, and I accept the metaphor there, is accurate up to about 1 in 10 to the 4. What is the accuracy when the cell actually divides and provides 2 new cells? It's 1 in 10 to the 10. Hardly a single, that's rather like a proofreader of 10,000 books, going through 10,000 books and making sure there's not a single error in the whole 10,000 books. How is that achieved? It's utterly amazing. It's achieved by the living cell. Because what then happens as the problem of the breakages, as we might call them, in the DNA formation from the double helix when it's unwound, what then happens is a whole army of enzymes go in and literally proofread the mistakes. And I only know, I mean, that's why I say you'd have to put my genome in 10,000 years hence into a living cell to do it.
I think the basis for self-non-self discrimination is this genetic origin, that we go by the code, the DNA, and then we decide different DNA, different species, self, non-self. So that's one of the, I think that's problematic. Now that we know that we as individuals and as humans, we need others to be healthy. We need all these bugs, we need other viruses and things to live within us. Another dimension to this, which I think is intriguing, is that what makes us different from one another? Our immune systems are incredibly heterogeneous. Everyone in here could leave a blood sample and then come back 6 months later and leave another blood sample, and it'd be very easy to pair them up, which one came from which individual, by just looking at the immune system and its composition. And if we think about what shapes us with respect to our immune systems, it's not the DNA. The DNA explains a very small fraction of that, but instead it's the experiences that our immune system has had during development and during time. So we are shaped by the environment in which we live, and consequently our self is changing. We don't have the same self when we are newborns that we do when we are 5 years old, or 10 years old, or 100 years old. So clearly, we need to shift sort of this understanding of what the immune system is doing from a view of discriminating self from non-self and rejecting everything that is genetically non-self, to an understanding of what the role of the immune system actually is, which is to maintain some kind of healthy balance. In the midst of this chaos in which we live, it's the immune system's job to make sure we stay on course, we stay healthy, in balance with everything around us.
From a biological perspective, we know that the female body is not the same before it's ever had children versus after it's had offspring, right? Mm-hmm. So we know that even from mice or other organisms, like the female form is different. When unfortunately, we still use the term virgin, but that is correct, technically correct. So those kinds of changes happen. There's some really interesting stuff to show that when females are pregnant, they will have the DNA of their offspring, like little fragments of it, in their own bloodstream, that their immune systems are substantially changed by the offspring that they have. It is quite interesting when you think about sort of biological individuality. Where does that boundary lie? Because there's different sort of levels at which you can draw the line. So when you were talking, my first immediate answer to is there any question about, or query about biological identity, my first impulse was to say no, because everybody has a genome. Like, we're taking a very human-centric conversation here, but every organism has its genome. Any sexually reproducing organism, let's say, to be very precise, has its individual genome. And so from the moment that that single-celled embryo is there with its set of DNA, essentially it is an individual. And you could argue that individuality comes from the fact that all the cells in your body contain the same genome. Mm-hmm. So the funny thing about science is that I will feel compelled to tell you that as being almost 99% correct, but also scientifically correct is the statement that not all cells in your body contain your genome. And also your genome does get altered through time. So even though, for example, I came from a single-celled embryo and there was one genome, and the majority of the cells in my body now do all carry that same sort of set of DNA recognizably. They've been knocking around for a few years, these cells and these bits of DNA. And so they will have changed just slightly from each other. And so the moment you open the door to that, you end up sort of inadvertently opening the door to the rest of the well, are you an individual in that case? Really?
I think of fantasies projected onto genes, and I think they've led to an enormous distortion of priorities in biology. I do have a slight vested interest here because in 2009 I took part in a debate with Lewis Wolpert, who's a leading materialist and mechanist at Cambridge at the Science Festival on the Nature of Life. And Lewis said, he said, it won't be long before, given the genome of a baby, of a fertilized egg, we'll be able to predict every detail of the ensuing child. And I said, Lewis, how long do you think that's going to take? I said, you said soon. I said, what, 10 years? He said, well, not 10 years, 20, no, well, maybe a century. And I said, well, that's a really, really long time. This is promissory materialism in an extreme form. I said, can we have, I bet you that it won't be possible even with a simpler organism within 20 years. And he said, well, I'm sure it will. So we have a wager. It was published in New Scientist. The one thing we could easily agree on was the stake. We wanted something that would increase in value over 20 years rather than decrease, like money. So it's a case of fine port. We paid half each. It's in the Wine Society vaults. So the wager is that by May the 1st, 2029, Wolpert said, given the fully sequenced genome of a fertilized egg, we'll be able to predict every detail of not a human, but at first he said, a chick. So I said, okay, Lewis, a chick. So then he rang me a week later and said, well, I've discovered a chick would be a bit too complicated. It'll have to be a frog. So I said, all right, a frog, a chick or a frog. And then after a week, he rang me and he said, I think even the frog's a bit too complicated. So it's going to have to be a nematode worm, Caenorhabditis elegans. And I said, well, that's a big step down. I said, well, if we're going to a millimetre-long worm, then at least we should spell out that to do this, you've got to predict the structure of every protein based on the primary structure of the protein, the sequence of amino acids, the threefold folded up structure. And he got back to me about a week. He said, can we just miss out the protein folding thing? I understand there's a bit of a problem predicting the tertiary structure of a protein from the primary structure. I said, yes, there is. I mean, that's the reason why this isn't going to work. And so he said, well, anyway, let's just leave it open. Can it be any organism? So the wager is by 2029, it should be possible to predict on the basis of the genome the structure of any organism, any multicellular organism. Well, that's the wager. And he left out epigenetics and all sorts of other things. Well, by May the 1st, 2029, we'll find out who gets the case of port.
So, of course, these systems are designed in a very different way. We have an outside-in design, and because computers, they're stabilized by our subset that we built deterministically. So it's behaving in a very fashioned way. We know what it's going to do in every moment. And the training is decoupled. We give it static data. It's not interacting with the world while it's training. It is based on a prediction paradigm, and it uses machine learning training algorithms. And our mind is very different. Our brains are using an inside-out design. It's self-organizing. It's coupled to the environment in real time. It's optimizing for coherence. And it's continuously developing itself. So technological design is this outside in, where you have a desk, where you understand everything, you understand all your tools, and then you are extending this by building an additional mechanism that extends your desk and gives it more functionality. And in nature, this is the other way around. You have a seed, for instance, the seedling of a tree that is by itself not a tree yet, but it wants to become a tree. And to do this, it needs to colonize a chaotic environment, subdue it, turn it into something that it understands that it can work with. And one way of doing this is it splits into lots of units that behave pretty much the same, so they become predictable, and then it creates some kind of scalable colony that is becoming larger and larger. And this is how this inside-out design works in nature and also in our own mind. If you look at the individual neuron, these are mouse neurons, embryonal ones, you see they are all single-cell animals that try to survive, each of them self-directed. And they have to link up and find an organization among each other that works, or otherwise they will starve to death. And this is how it works. It's completely inside out. And so there was a branch in computer science that looked at these principles of self-organization. So far it didn't get really far, but it already started with Turing, who looked at reaction diffusion patterns, chemical reactions that give you regular patterns that can be dynamic, and looked, can we use them to understand computation and self-organizing systems? And then some people like Alex Mortwintsev, who's working at Google in Zurich, build simulations that are more modern, that try to generate such patterns in a general case. And then it comes this idea of cellular automata by Conway and others, where we build computation in a systematic way out of simple systems that only look at the state of their neighbors, of their environment, and then change state accordingly to what they observe in their environment. And the biologist Mike Levin is working with this, extending this into neural cellular automata that explain development in organism, but also possibly the self-organization of our brain. And so we can already take this to learn, for instance, textures and so on. What would be really interesting is to try if you can learn arbitrary functions to control behavior with such self-organizing principles. And the neuroscientist Jerry Edelman suggests that our genome is not encoding a very specific architecture for our brain. Instead, in everybody's mind, there is an evolution going on, an evolutionary competition between different forms of governance. And there is only enough information in the genome to bias that competition, so it converges relatively quickly into the right architecture. So every mind is an evolving system. Everyone has an individual evolution in our mind for structure.
And then this line of thinking leads to modern thinking in transhumanism and posthumanism, that maybe we want to outsource part of our brains. I'm certainly outsourcing a fair bit of my thinking to this little smartphone. Why do I need to know where things are when I can just get a map? And as Nick Bostrom describes in The Future of Human Evolution, one scenario might be that we outsource more and more. We outsource language abilities, math abilities. Why should I have my memory in my head when I can have it in the cloud? And in the end, we end up with a civilization where there is nobody around. There is a great economy. There is a lot of technology. Every curve is pointing upwards. The only problem is, of course, that there is nobody around. So in this case, you might have a scenario where we, either because of pursuit of pleasure or pursuit of effectiveness, end up with a civilization that has actually lost all value. And Scott Alexander, in his brilliant Meditations of Moloch, suggests that this might be a common failure mode for many different scenarios. And wireheading is just one of them. Indeed, it has been proposed as a solution of the Fermi paradox. Why aren't we seeing any aliens? And maybe it's possible that progress undermines itself because at the end, you don't need to do very much. You can live in your utopian world, and then you have inner bliss. And at that point, there is no point anymore in looking outward or sending messages. And indeed, some people are suggesting that maybe it's obesity that is the problem. So we might be having that all sufficiently advanced civilizations solve many of the problems, but the evolutionary demands, like getting food, then make them do maladaptive things, which means that they're actually having trouble doing anything else. Now, this is not just a problem for humans and maybe for civilization. It might also be a problem for artificial intelligence. So this is a classic example of having an artificial intelligence program learn how to play computer games. And the goal here is to maximize the points. And human would normally play this game by trying to get around the course with the boat and then win by getting around as fast as possible. However, those little green things give you bonus points. So it's smart if you're a human to get a few of them, but you want to get into a goal. However, the program is only trying to maximize the score and found that endlessly, forever, circling around like this maximizes the point. This is totally missing what we would say is the point of the game, but to the artificial intelligence, which isn't that intelligent, it's of course exactly what it wants to do. So this actually is a generic problem. It turns out that software quite often misbehaves, even when you give it a goal it's trying to achieve. In the early 80s, Lennart's Erisco program got punished in that sense for doing errors. And it figured out that if I just shut myself down, I'm not going to get punished at all. There are various other interesting systems where you find that they are misbehaving quite wildly and doing quite creative things that maximize what we told it to maximize, but not what we actually think is the point of playing the game. Now, this is a bit of a problem, because when you start thinking about using robots in the real world, autonomous cars, or having software regulate our stock market or our infrastructure or our nuclear arsenals, we better get the reward function right. But many of these systems turn out to be very good at finding ways of short-circuiting that reward function, making it solve the problem that we gave it, but rather than what we wanted it to solve. And this can be pretty disastrous. Even more interestingly, it can do it in a highly active way. Our picture of the indolent wireheader that is just sitting around in total bliss can, in the case of a robot, be something that is rushing around, reorganizing the world in order to maximize its own reward. So this has led to work on abstracting wireheading, essentially linking it to the concept of Goodhart's law in economics and sociology, that if you have a measure of something, you tend to maximize the measure rather than the thing you try to maximize, which is why we get grade inflation in schools. We want to maximize knowledge among the pupils, but unfortunately we measure that using grades, so everybody tries to get good grades. And the problem here is, of course, that we are very bad at actually getting the goals right.